And we will go. Welcome everybody to this month's Centers in Iron and Heme Disorders virtual seminar series. We're so happy to have you join us today, and we'll hope that others join in. Before I let Dr. Bowden take over to introduce our guest speaker, I want to just share my screen and remind you of a couple of opportunities for funding. Let me just find, first of all, well, that's not the one I wanted, but okay. So I want to take you to our home website, the CIHD's website. And if you watch here in the next couple of weeks, there will be an opportunity, a call for funding for U54 pilot and feasibility. This just shows people who've received them in the past. But if you watch on our website, there will be an announcement for new pilot awards that can be submitted. The call date will be sometime in late June, probably, for the due date. And these run up to about $50,000 for a year of funding. So I hope you will take the opportunity to go in there and look. The other place I want to call your attention to is the overall Centers of Excellence in Hematology home website. So this is the website here. If you click on funding opportunities here, what you will find is that there's a new call for Type B applications that's due in September. These are up to $75,000 for PNF funds, and you can read the details here. There's also an opportunity to apply for what we call core credits. And you guys, this is wonderful. It's you bank money at one of the cores that's supported by the CCH, not necessarily Utah's, anywhere, and you can get up to $12,000 worth of core credit. So if you, for example, want to do iron and heme studies or metabolomics or mutation generation detection here at Utah, you can write for these funds and they get reviewed like every month. And the money it gets banked at the core, and you can use that core to generate your mutations, to generate metabolomics data, to generate imaging data, all kinds of things like that. So I highly recommend you go look here if you're looking for, developing some preliminary data for a larger grant proposal. And then I'm going to stop sharing there, and I'm going to turn the time over to Anna to introduce our guest speaker.
Wonderful. I'll make this relatively brief too, so we can get on to Daisuke's wonderful work. It is my pleasure to introduce Dr. Daisuke Nakada, who is an associate professor at Baylor College of Medicine. Dr. Nakada has been at Baylor. How long have you been at Baylor?
11
years. Oh, wow. Okay, wonderful. Before that, he was a postdoc at University of Michigan with Sean Morrison, where I, as a postdoc, remember reading his work on estrogen-driven extramedullary hematopoiesis that I was fascinated by. His work, one of the things that I love about Daisuke's work is how broad it is. It encompasses so many different aspects of hematopoiesis, and primarily or really focused on regulation of hematopoietic stem cell biology, from metabolism, lineage bias, including erythropoiesis and megakaryopoiesis. He does genome editing. He does kind of a little bit of everything. He's funded, has extraordinary funding from NIH, from LLS, and his work has always just been such high quality and really field changing. So we're very excited to have you here, Daisuke, and to hear about your work today on hematopoietic stem cell regulation. Thank you so much.
Hey, well, thank you for the introduction. And let me share the slide. Okay. Well, it's a great pleasure to talk to you about our work. And today, I recognize that this is an NIDDK funded center, and I'll have to say at least half of the lab is working on malignant. And so I'm going to refrain from talking about malignant hematopoiesis, but just as an introduction, as Anna mentioned, we are interested in hematopoietic stem cell biology and what it takes to be hematopoietic stem cell, how they respond to stresses and changes in animal physiology, like Anna mentioned about pregnancy and stuff. And we're interested in how these hematopoietic stem cell mechanisms become dysregulated to cause malignancy. As I mentioned, a lot of the effort in the lab is now focused on leukemia, and especially leukemia stem cells, because we can draw multiple parallels between hematopoietic stem cells and leukemia stem cells in trying to understand, again, what it takes to be leukemia stem cells. But we're getting more interested in metabolism of leukemia stem cells, because we find many instances where there are unique metabolic vulnerability of leukemia stem cells that are not found in normal hematopoiesis or hematopoietic stem cells, such as those that listed down here with, let's see, I can get a laser pointer, like AMPK, more recently about selenium that we found that is really essential for leukemia stem cells. Now, I'm not going to talk about that today. Another area that we are interested in, we're starting to get interested in, is clonal hematopoiesis. And I don't think I need much introduction about CH, but this is a phenomenon in which mutant hematopoietic cells, likely hematopoietic stem cells, accumulate with age. And we are particularly interested in the environmental factors that leads to CH. And if you think about the exposures, those are, for example, infection, inflammation, chemotherapies, radiation, and those are all the tools that we as a field use to study hematopoietic stem cell response to these stresses, how robust stem cells are, how they regenerate hematopoiesis after these stress. So the field of clonal hematopoiesis is starting to remind us all the tools that we have been using, and sort of ways to revisit what we have found in hematopoietic stem cells, and maybe we'll find new biology of clonal hematopoiesis. So just a brief couple of slides about clonal hematopoiesis. As all of you know, it's a phenomenon in which mutant hematopoietic cells accumulate with age and clonally expand, as you can see from these yellow cells. It clonally expanded with age, and recent studies has found many health implication of CH. This is one of the probably the most iconic figure in hematology now, done by Sid Jaiswal. The incidence of clonal hematopoiesis increases with age, and when we are in our 70s, maybe 10% of the people with 70s, depending on the sensitivity of the sequencing, carry detectable clonal hematopoiesis, and the incidence of CH increases with age. The mutations that are found in clonal hematopoiesis are the recurrent mutations that are found in acute myeloid leukemia. And in particular, with CH, the top 3 genes that has been found are so-called the DTA mutation, GNMT3A, TET2, and ASXL1. And because CH is a function of age, long-lived animals like humans are likely to be impacted more than short-lived animals like mice. And recent studies has found that the mutation rate between very short-lived animals like mouse and humans vary dramatically. For example, mice that live only 2 years accrue hundreds of mutations a year. And by the end of mice 2-year lifespan, maybe those animals accrue a couple of thousand mutations per cell. Whereas humans that live up to 80 years and even more accrue much fewer mutations, maybe tens of mutations a year. But by the end of the life of human, maybe we are accruing almost the same amount of mutation as mice. Also depicted here, mice live shorter, but the end of life mutation burden between mice and human or different animals seems to be about the same. Now, the implication of this is that we humans are actually giving quite a long time for those clonal hematopoiesis clones to grow. And another implication is that we have to deal with those mutant clones for decades compared to mice that might deal with it for 1 year or something. So it's certainly a more significant health impact on humans. As we all know, clonal hematopoiesis, because it's almost the first hit in heme malignancy, it increases the risk of heme malignancy significantly. And a recent work by Ben Ebert has found multiple risk factors such as age, RDW, or particular CH mutations that have high risk for developing leukemia, such as spliceosome or JAK2. And by combining those, they can actually predict quite significantly, like 50% chance of developing AML in the next 10 years. Those are the CH people with those multiple high-risk factors. And in addition to myeloid malignancies, CH carries risk of cardiovascular disease, pulmonary disease, osteoporosis, liver disease, and many other diseases that often associate with metabolic disorders, but some of them don't. Another risk for myeloid and heme malignancy that has been known for quite some time is actually sex. Has been well known that male have much higher risk of developing heme malignancies. Shown here is AML and MDS. Male have much higher risk of developing these diseases compared to females, as you can see here, and especially notable in MDS. Now, this is not only limited to these particular two heme malignancies, others such as multiple myeloma and so on also have a sex difference in the incidence. And that started to lead to our research on studying sex difference in hematopoietic stem cells. Now, the mechanism by which male have higher risk of heme malignancy remains somewhat unclear, but a recent study has shown that there are particular mutations that male tend to accrue compared to females. The tables, the figure here shows all the mutations. Some of the mutations are on the sex chromosomes, like KDM6A, ZRSR2 are on the sex chromosome, but there are some mutation on the autosomes, such as ASXL1, RUNX1, TET2, SRSF2, U2AF1, that have a higher, that males accrue more compared to females. For example, here, U2AF1, much higher incidence of mutation in male compared to female. And if you remember those who know about CH will realize that, oh, these are actually some of the high-risk CH mutations that leads to heme malignancies. Another important take-home message from this study is that there weren't any sex difference in late leukemic events, or so-called the secondary mutation that leads to activation of signaling pathways such as RAS or tyrosine kinases. Those weren't necessarily showing any sex difference. And this implies that if there were to be a mechanism by which males tend to get more leukemia, it is likely to be happening at the clonal hematopoiesis level or pre-leukemic hematopoietic stem cell level. Now, is there any sex difference in clonal hematopoiesis? Another recent study showed that although the overall incidence of clonal hematopoiesis is not significantly different, in fact, the authors mentioned that females exhibit a slightly earlier incidence of CH than male. But the striking difference was in the types of mutation that male and female accrued. There was some mutation that were more frequent in females, such as GNB1 and DNMT3A. By the way, DNMT3A has been shown to be a lower risk mutation in terms of developing heme malignancies, whereas male tend to develop other mutations such as SRSF2, ASXL1, SF3B1, and JAK2, and several others. And again, these tend to be the ones that carry higher risk for heme malignancy, especially the spliceosome mutation as well as JAK2. This is highly significantly as a risk factor. And this raises the question of, are there any sex difference in hematopoietic stem cell regulation? Now, this is somewhat of a retrospective asking of a question, because we started the study that I'm going to mention years before these clonal hematopoiesis findings. But these recent findings about sex difference in mutation in clonal hematopoiesis as well as acute myeloid leukemia is making us revisit what we have found in these sex difference in hematopoietic stem cells that we found before. So several years ago, we were interested in it's basically a fundamental question: are there any sex difference in hematopoietic stem cell function? And they were present about sex difference in immunology. For example, adaptive immune system will tend to be stronger in females than male. And so there was some indication that there will be some sex difference in HSCs as well. But at that time, there was not a clear evidence for that. So what we did is to go to mouse models that we use, regular black 6 animal, and injected BRDU for 10 days and did a basic BRDU incorporation assay in hematopoietic stem cells. And what we found is that over this 10 days of BRDU incorporation, we found that female hematopoietic stem cells incorporate more BRDU than male hematopoietic stem cells. And you'll notice that this is a significant but a small difference, and it's even small if you consider that it has been 10-day period. And so day-to-day difference is very, very subtle, but it can accrue over time over 10 days. And we confirmed this by H2B GFP dilution assay. So it's nothing, we don't think this is something particular about BRDU metabolism difference between male and female. We believe that this is a difference in hematopoietic stem cell division difference. So what is the mechanism that regulates the sex difference in hematopoietic stem cell division? Now, there are several things that we can think about. One could be genetics. It could be the difference between XX chromosome versus XY, or it could be endocrine system. And so we took the latter approach, and we did castration and removed the testes. That did not affect hematopoietic stem cell division of male mice. But when we did the ovaryectomy surgery and removed the ovaries, we found that that significantly reduced the division of female hematopoietic stem cells, demonstrating that it is the endocrine system that is talking to hematopoietic stem cells in females and driving them to divide more. If it's estrogen, if it's ovaries, is it some sort of sex hormones? And a long story short, we found that estrogen receptor alpha was unique among several hormone receptors in a way that it is highly expressed in hematopoietic stem cells and not in other hematopoietic cells. So we decided to delete ER alpha from hematopoietic cells. And what we found is that, as shown before, there is a sex difference in BRDU incorporation in HSCs. You can see female incorporate more. But when we delete ER alpha from the hematopoietic system using MX1 Cree, we lose that sex difference. And we confirm that in this MX1 deletion system, there's no change in the endocrine system. Estrogen is still produced in such females. And so this demonstrates that ER alpha is the receiver in the hematopoietic stem cells to respond to estrogen receptor to estrogen. Now, I don't have time to talk about this, but the significance of this at that time was that we proposed is during pregnancy when estrogen level increases in a physiological manner. And we found that if you delete ER alpha from the hematopoietic system, we ablate some of the changes or hematopoietic changes that we find during pregnancies. Now, after this study, a grad student picked up this project, Richard Chappell, and asked the question, is an increased division of hematopoietic stem cells by estrogen, does it have to do anything with the regeneration? And before we started this project, there were studies showing that hematopoietic stem cell division is generally negative for regeneration. For example, if you isolate hematopoietic stem cells that are already in cycle, such as SG2M, and transplant those stem cells, they tend not to do well in transplantation assays. So one possibility was that estrogen drives hematopoietic stem cell division. Maybe if we transplant those cells, they don't do well. They don't regenerate the hematopoietic system. Now, we were actually surprised by the finding, as shown here. We found that compared to hematopoietic stem cells isolated from animal that treated with oil as a control, it regenerated the blood system normally. But if we take stem cells from mice treated with estrogen, they actually regenerate, especially the myeloid lineage, myeloid cells and platelets and red blood cells, better than those that were treated with oil in vivo. And this beneficial effect on regeneration was ablated if we delete ER alpha from those animals. So it is a mechanism which ER is really talking to ER alpha in HSCs and improves the regenerative potential. In terms of mechanism, we did RNA sequencing analysis of those stem cells that were treated with oil versus estrogen, and some of the suspected candidates such as MYC and E2F, because those stem cells are dividing more often, such as also OXPHOS because they're actively dividing and metabolically active. Those pathways were upregulated by estrogen. But one of the pathways that we were interested, because it was somewhat unexpected, is the unfolded protein response that was increased after estrogen treatment. And so what is the unfolded protein response? It actually consists of 3 branches, one governed by a protein called IRE1 alpha, another one called PERK, another one called ATF6. But the goal of this, the combined pathway is to, one, improve the protein homeostasis by upregulating chaperones or foldases, try to maintain protein homeostasis. Another possibility of the outcome of this activation of this pathway is to basically kill the cells through apoptosis when the protein homeostasis is so compromised. And one of the branches that we found activated in estrogen-treated hematopoietic stem cells is governed by this protein called IRE1 alpha. And as shown here, based on FPKM of RNA-seq, estrogen-treated hematopoietic stem cells significantly upregulated ERN1, which actually encodes for IRE1 alpha, whereas this response was not seen in hematopoietic stem cells that were deficient in ER alpha. So it is an ER alpha dependent transcriptional mechanism. We found that using ATAC-seq, there is a potential ER alpha binding site on the promoter or upstream of IRE1 alpha gene, as shown here, and has a pretty significant estrogen response element consensus site. And when we do a reporter assay using this construct, we find that the transcriptional response is activated by expressing ER alpha and treating with estrogen, shown here. And if you make point mutation in this ER alpha binding site, we no longer see this luciferase activation. So this site appears to be the factor that responds to estrogen and ER alpha. And indeed, when we do CHIP PCR with isolated hematopoietic stem progenitor cells treated with estrogen, we find that ER alpha indeed binds to this peak we also call P2 in this diagram. So ER alpha seems to be directly binding to this site, increasing the transcription of IRE1 alpha. So is IRE1 alpha really doing something in hematopoietic stem cells? To address this, we used a CRISPR-Cas9 system and deleted IRE1 alpha or the ROSA26 locus in purified hematopoietic stem cells, transplant those cells into recipients, treated the recipient with oil or estrogen, and quantified hematopoietic stem progenitor cells. And what we find is that, consistent with the function of estrogen in promoting HSC division and regeneration, if we just delete control ROSA26 locus and treat with estrogen, we see increased hematopoietic stem progenitor cells in the recipient animal. Whereas if we delete IRE1 alpha, we no longer see this beneficial effect of estrogen, indicating that estrogen requires IRE1 alpha to exert its pro-regenerative function on hematopoietic stem cells. So just to summarize this part, we found that ovaries, or the endocrine system governed by ovary, regulate the rate of HSC division via estrogen. Estrogen also regulates the regenerative potential of hematopoietic stem cells, and estrogen ER alpha transactivates IRE1 alpha, and IRE1 alpha is required for estrogen to enhance HSC regeneration. Now, I mentioned that kind of the new discoveries in clonal hematopoiesis and sex difference is starting to make us revisit this, revisit these findings. And at this point, we don't have any solid evidence to suggest that this mechanism governed by estrogen is responsible for clonal hematopoiesis. But at least we can, speculate and have some discussion about it. And one possibility is that to explain why there could be clonal hematopoiesis difference in between sex is obviously estrogen could be anti-inflammatory. A lot of the clonal hematopoiesis findings evolve around inflammation, and mutations such as DNMT3A, TET2, JAK2. Actually, the expansion is exacerbated by inflammatory stimuli, IL-1 or IL-6. And it is well known that estrogen is anti-inflammatory. So maybe by the fact that estrogen is more abundant in females, is less inflammatory, that might change the way in which some clones might expand or not. But it doesn't really explain why there's particular mutations.
Oh, Daisuke, I think you got muted.
Yeah, I'm not sure what happened there,
Everyone got muted somehow.
but not you and me. Ask to unmute. Did it? What?
Okay,
You're back.
Okay, you're
Okay.
back.
Am I
That was very
still
strange.
sharing?
Yeah, you stopped share.
Okay.
So weird. Okay.
Is this?
We lost you just like 30 seconds ago.
Am I back on?
Yes,
Okay, great.
sorry.
So another possibility, a little bit more provocative, but is the enhanced HSC division by estrogen leveling the competition between CH and non-CH hematopoietic stem cells. Now, as I mentioned, there are a lot of environmental factors that drive or select mutant hematopoietic stem cells, such as TET2 cells are selected for by inflammation. And maybe if every hematopoietic stem cells are dividing at a higher rate, it changes the competition dynamics between mutant and non-mutant hematopoietic stem cells. And finally, another possibility is estrogen might be negatively acting on some male-dominant CH mutations, such as spliceosome. And I think particularly interesting is the IRE1 alpha, or so-called integrated stress response, that others in the field have found that it can affect the HSC survival, and maybe that pathway is somehow acting on mutant hematopoietic stem cells. Now, we are working on this, and I hope to be able to talk about this in some near future. So I'm going to switch gear and talk about a different story about erythropoiesis and hematopoietic stem cells. And during the course of the previous study, we noticed that there is a gene called keratin 18 that is specifically expressed in hematopoietic stem cells, as you can see here from the gene expression common data set from Seita and Weissman, expressing hematopoietic stem cells, but very low in other hematopoietic cells. Also by our own quantitative PCR assay showing high level of expression in hematopoietic stem cells. And keratin 18 is not a very known gene in hematology, but it's actually one of the important marker for skin stem cells, as well as prostate stem cells and progenitor cells in those tissues. So we decided to use this keratin 18 CRE-R system that was developed by Cedric Blancpain to study skin and prostate, and use this to see if we can label hematopoietic stem cells. Because around that time that we worked on this, there wasn't very much known about the physiological contribution to hematopoietic stem cells. People relied on transplantation to assess their function, but in a transplantation assay, hematopoietic stem cells inevitably contributes to all hematopoietic cells because all of those cells are depleted by radiation. But the question that we can ask with these lineage tracing is, what are hematopoietic stem cells doing on a day-to-day basis? So we obtained this model and labeled with tamoxifen. And what we found is that, as I mentioned, it is fairly highly expressed in hematopoietic stem cells, but not in other cells. So if you analyze shortly after tamoxifen injection, we see pretty nice labeling in hematopoietic stem cells, but much less or almost and undetectable in some of those mature cells. And if we chase this over time, 4-week, 26-week, and almost a year, we find that many hematopoietic cells start to accrue or gain YFP positivity, indicating that the YFP positive hematopoietic stem cells gradually contributes to hematopoiesis over the span of 1 year. Now, this is fairly a slow response.
Sorry, Daisuke, I missed it. When are you initialing tamoxifen labeling in this experiment? What age? 12 weeks or 8 weeks?
These are around 2 to 3 months old.
Okay, so 8 to 12 weeks.
8 to 12,
okay.
yeah. Young adult,
Okay, cool.
yes.
Thank you.
So this is a fairly slow response, but what we do find is that platelets is one of the lineage that receives significant contribution from hematopoietic stem cells. And this is actually something that multiple groups agree upon. Fernando Camargo's group also find platelets is one of the major contribution from hematopoietic stem cells. Now, what happens if you stress these animals, almost like a transplantation assay? So what we did is treated the keratin 18 YFP animals after tamoxifen with 5-FU and waited 3 or 4 weeks. And what we found is that regardless of this time point, we see increased contribution of hematopoietic stem cells to different lineages, especially platelets, as you can see here, control versus 5-FU, strong contribution to platelets, as well as myeloid cells. But still, the lymphoid cells receive much little contribution. As you might predict, that the lymphoid development takes quite a long time. So this is the model that now allows us to really study how stem cells respond to different kinds of stresses. And 5-FU was nice that it ablates many of the cells downstream, such as myeloid, platelet, red blood cells, and also some impact on the lymphoid cells as well. But the question that we next became interested in is what happens if you just ablate single lineage? Do stem cells respond to that by generating just that lineage? Or do they just respond in a generic way and make all kinds of cells, as you might predict based on the capacity of hematopoietic stem cells to generate all kinds of cells? So the question is, if we deplete red blood cells, are they going to respond, stem cells going to respond to this and just make the erythroid cells and regenerate the erythroid lineage, or are they just going to make all the cells? And so this project was done by former grad student Nina Tsen, and she used one model of erythroid stress, which is hemolytic anemia induced by a chemical called phenylhydrazine. And so what she did is treated these animals with tamoxifen to activate in this case, TD tomato, in the same keratin 18 CREAR system, and then treated with 2 doses of phenylhydrazine and analyzed 2 weeks later. As you can see, phenylhydrazine causes really robust changes in the spleen size, splenomegaly, as you can see here. And when we looked at the bone marrow hematopoiesis, we did not see any changes in the contribution from hematopoietic stem cells to these different cells, including intermediate progenitors such as GMP, CMP, MEP, as well as the mature myeloid cells as well. Now, the picture was quite different when we looked at the spleen as well as the peripheral blood. First, we found that red blood cells was the only lineage in peripheral blood that had increased contribution to from the hematopoietic stem cells and not platelets or B and other cells. In the spleen, we find that multipotent progenitor cells, which we consider as the immediate downstream of hematopoietic stem cells, received significant input from hematopoietic stem cells, as well as MEPs, megakaryocyte erythroid progenitors, as well as red blood cells that were present in the spleen. So as we somewhat predicted, hematopoietic stem cells do respond to erythroid stress, and have a specific response in a way that it promotes the erythroid output and not a generic output to different kinds of hematopoietic cells. And one of the what we find interesting was that the spleen seems to be a unique organ in which this erythroid response is happening. And so when we look at the hematopoietic stem cell numbers in bone marrow as well as in the spleen, again, we don't see any changes in the numbers of hematopoietic stem cells in the bone marrow after PHC treatment. But in the spleen, we see a significant expansion of splenic hematopoietic stem cells. And about 7 days after PHC, which I mentioned, when the spleen size normalizes, the number of hematopoietic stem cells also approximately normalizes. Are those stem cells in the spleen functional? And so to address this, we did a limiting dilution transplantation assay with different doses of either bone marrow cells or splenocytes. And the conclusion is that with PHC treatment, there's really no difference in the frequency of functional stem cells in the bone marrow, either 1 in 35,000 or 1 in 52,000. But in the spleen, there was a significant expansion, approximately 5-fold increase in the functional hematopoietic stem cells. In control, it was 1 in 3 million, and in PHD, 1 in 600,000. So approximately 5-fold increase in functional stem cell frequency by hemolytic anemia in the spleen. We found that spleen is unique in a way that those stem cells in the spleen have much activated cell cycle after PHC, as you can see here, as well as in the MPP. And we don't see this dramatic activation of stem cells in the bone marrow. Rather, we see somewhat more of a quiescent stem cells remaining in the bone marrow. So the model of PHC, sometimes we get criticism. It's basically a poison. It could cause inflammation. And so another model to address this erythroid stress is to do a serial bleeding. And so we modeled based on an earlier paper from Irv Weissman. We did 3 days of bleeding and then immediately transfuse either red blood cells or PBS. And what we find is that if we do this serial bleeding and without transfusion, we see a significant increase of splenic hematopoietic stem cells, similar to what we find with PHC. But if we transfuse red blood cells back right after bleeding, we substantially rescue that, well, mitigate that stem cell expansion, whereas transfusion of a blank PBS does not have that effect. So similar to what Irv has concluded years ago, hematopoietic stem cells may have the ability to sort of respond to changes in red blood cells. And so next we did a transplantation of purified hematopoietic stem cells from bone marrow or spleen after PHC treatment. And the conclusion here is that controlled splenic hematopoietic stem cells, as shown by this red line, have a not so great potential compared to bone marrow hematopoietic stem cells shown in this black line here. So the function of hematopoietic stem cells is somewhat impaired compared to bone marrow hematopoietic stem cells. But if we isolate splenic hematopoietic stem cells after PHC treatment, we see a significant improvement or much stronger output than regular splenic HSC shown by this blue line. And this was particularly notable in myeloid, red blood cell, and platelet lineage. So PHC seems to be affecting the myeloid and erythroid lineage strongly in the splenic hematopoietic stem cells. In terms of the mechanism by which hematopoietic stem cells respond to phenylhydrazine and hemolytic anemia, we focused on iron. We used a dye called calcium AM, which shows an inverse. So when cells have higher contents of iron, they lose fluorescence, as you can see here. So what we find is that if we compare control spleens of day 3 or day 5 of PHC treatment, we see increased iron contents as shown by the reduced calcium AM staining. So with stem cell expansion, stem cells are accruing more iron content. And this is consistent with the increased expression of CD71, which is the transferrin receptor. Without phenylhydrazine, we see very little CD71 positive fraction in spleen HSCs, but with PHC treatment, we see a significant expansion of such population. And so if we see increased iron, can we change the hematopoietic stem cell function by chelating iron? So we use a drug called DFO, which is the iron chelator. And iron chelator indeed works because if we, as I mentioned before, PHC increases iron contents as shown by the reduced calcium AM staining. And if we chelate with DFO, we now normalizes this change in iron content. As I mentioned, phenylhydrazine increases the splenic HSC number, and this is also rescued by DFO. And also the division of splenic hematopoietic stem cell increases PHC, and that is also rescued by chelating iron. So iron regulates the hematopoietic stem cell expansion in the spleen as well as its stem cell division in the spleen, but not, as I don't have time to show, but it doesn't affect the bone marrow hematopoietic stem cells. With the single cell RNA sequencing of the stem cells in the bone marrow and the spleen, and we find population that is consistent with hematopoietic stem cells, myeloid progenitors, erythroid progenitors, and some lymphoid progenitors. And so how are they different between control and PHC treated tissues? So in the control bone marrow, we see nice enrichment of hematopoietic stem cells, very little population that we call them erythroid progenitors. But in the PHC-treated spleen progenitors, we see this expansion of population 3, which we consider them as erythroid progenitors, as well as this population, which we think are immature erythroid progenitors upstream of this population. This population express the well-known erythroid markers, such as KLF1, GATA1, and transferrin receptor. As you can see here, the population that expanded upon PHC indeed expresses much higher level of these genes compared to the bone marrow and other hematopoietic stem progenitor cell population. Now, we were intrigued by this paper published by Dan Landau, establishing that erythroid transcription factor binding sites are enriched for CPG sites, and accessible chromatin sites with erythroid transcription factor motifs become hypermethylated in TET2 knockouts. So in both mice and humans, some of these transcription factors that are known for its role in erythropoiesis have much higher CPG frequency than transcription factors that are well known for their myeloid potential. And these transcription binding sites become hypermethylated in TET2. So we wondered whether TET2 is the underlying mechanism that regulates these erythroid changes in hematopoietic stem cells after hemolytic anemia. And so we obtained the TET2 knockout, the conditional knockout using MX1-CRE. We treated them with PHC and transplanted them and assayed erythroid progenitor regeneration. When we transplant wild-type HSCs treated with PHC, we see this increased capacity of those cells to regenerate erythroid progenitors, but this potential was lost if we delete TET2. Also, some of the other progenitors that are immunophenotypically identifiable, CFU-E and Pro-E, they were expanded in wild-type spleen after PHC treatment, but that expansion is not seen in a TET2 knockout animal. So what is TET2 doing? What we found that TET2 is not activated in terms of transcriptional level, but we find that TET2 protein is accumulating and increased in PHC-treated spleen hematopoietic stem cells. So these are single-sorted hematopoietic stem cells from animals either treated with control vehicle or PHC. And as you can see here, TET2 is more abundant compared to control bone marrow hematopoietic stem cells, also quantified here. Now, there had been several studies about TET2 regulation at the translational or post-translational, to be post-translational step. And there had been several mechanisms by which it is degraded, such as calpain-mediated or caspase proteasome and autophagosome leads to TET2 degradation. And this is one area that we are actually actively working on, because we think that it's pretty significant in the context of clonal hematopoiesis as well. But what we find is that when we isolate hematopoietic stem cells from mice and treat with several of those inhibitors that inhibit those candidate mechanisms, we found that calpeptin is actually unique among others that can suppress the TET2 degradation. So this is a cyclohexamide chase experiment. And so normal HSCs lose their TET2 within 4 to 6 hours. But if you treat them with calpeptin, you can retain a significant amount of TET2 as quantified here. But this stabilization is not seen by a caspase inhibitor. So we believe that calpain is doing degrading TET2 in hematopoietic stem cells, and we are exploring whether this has to do with clonal hematopoiesis or other malignancies that relies on TET2 loss. So in the last couple of slides, I want to just show that hematopoietic stem cells depend on TET2 for increased erythroid gene expression. So if we take PHC-treated splenic HSCs, they exhibit increased expression of several of these erythroid genes, such as transferrin receptor, and we don't see that response if we treat TET2 knockout with phenylhydrazine. So molecularly, we find that at least for transferrin receptor, we think that there is a site that is demethylated by TET2. And so when we analyze 5-hydroxymethyl-seq data from others, we find peak that has several CPGs, especially strongly 5-hydroxymethylated in MEPs. And when we analyze this site by bisulfite sequencing, we find that splenic HSCs treated with PHC have a significant demethylation of the CPG sites, whereas if we do the similar experiment with TET2 knockouts, we don't see that demethylation. So at least for transferrin receptor, we believe that there are some sites that are demethylated by TET2, leading to increased expression of some of these erythroid marker genes. So to summarize the second part of the talk, we use HSC lineage tracing to show that hematopoietic stem cells have the capacity to respond to stresses that impair particular lineage. In this case, we showed with phenylhydrazine as well as serial bleeding as a way to cause erythroid stress. I don't have time to talk about, but we also see a very similar thing if we deplete platelets with the antibody. Stem cells respond to platelet depletion, and they specifically regenerate platelets, but not erythrocytes. Splenic HSC become activated and erythroid primed after anemia. And we think that that is through some mechanism by which stem cells are increasing iron uptake, and that is leading to erythroid priming. Through this iron mechanism, we think that TET2 is a critical downstream. TET2 is stabilized and is required for erythroid priming of splenic HSCs. And we find, at least in some of the genes, TET2 is involved in DNA demethylation of particular sites, leading to increased expression of those erythroid priming genes. So last, I want to thank the people who did the work. The first couple of experiments, the sex hormone stuff were done in the lab with Richard. Richard was the one who did the estrogen treatment and the regeneration study. The second part of the talk with erythroid stress was done by Nina and also Yuki. Work was done with collaboration with people around the Texas Medical Center. Jim Martin and Hassan Sami helped us on single-cell RNA sequencing and its analysis. Nancy Huang helped us on a TET2 analysis, and Koichi Takahashi on some of the methyl sequencing. And we were grateful for some of the funders that supported our work. And I'll be happy to take any questions.
Great talk. Questions for Dr. Nakada,
Can I ask a question?
please.
Can you elaborate a little bit? You mentioned you don't see any effect, any change also in the iron, in the bone marrow, in the stem cells, and so on. Why do you think your effect is primarily in the spleen? I mean, it's just the fact that you have most of the expanded erythropoiesis in the spleen in the mouse, and what would you think it's gonna happen if in a splenectomized mouse, for example?
Yeah, that's a great question. As you may know, so there's a lot of turnover of erythrocytes in the spleen in mice, and that might lead to local changes in iron handling in splenic hematopoietic stem cells. Splenectomy will be a really nice experiment to address that. What I would predict is they will probably move to somewhere else, like liver. Extramedullary hematopoiesis happens not only in the spleen, but also in liver and other tissues. So my prediction will be that the similar mechanism might happen in other tissues.
But you might force also the bone marrow maybe to show some changes, and maybe more transposable also to humans, to what happens. I mean, I think it would be cool to see what happens when you have a splenectomy.
Right,
and
yeah.
see you have some changes.
Yeah.
Thanks.
I think in terms of human relevance, there's recent studies by a group in UK that did human single-cell RNA sequencing with, I believe it was some disorder of erythroid disorder that found human splenic hematopoietic stem cells are also primed to undergo erythropoiesis. So I think,
I think we are underestimating that there might be something also human.
Right, exactly, yeah.
Thanks.
Bean, do you want to ask your question directly?
Sure. Actually, I've got two now, but I'll ask the first one. This is about the first part of your talk. So it's not like males have zero estrogen. They have aromatase in their peripheral tissues, and that can convert testosterone to estradiol. And strong expression is found in fat cells and bone and endothelial cells, which all sort of make up a bone marrow niche. So I think it's likely that there would be some low level of estrogen present in males in the hematopoietic niche. Do you have any sense of whether or not your ER alpha knockout males have more clonal hematopoiesis than wild type males? And would it be possible to knock out aromatase in some of those tissues and see if they get even worse clonal hematopoiesis?
Yeah, those are really the questions that we are starting to, what we will want to address in the next couple of years. In terms of ER alpha function in male, we really couldn't find anything in a steady state. Now, it doesn't mean that male cannot respond to estrogen. In fact, they strongly respond, even maybe stronger than female, to exogenous estrogen. And so they divide much strongly if you treat male with estrogen or estradiol. So they have the mechanism to respond, but I believe that just the physiological level is much lower, that we really don't see much effect in a physiological state.
Okay. And then given that you were talking about humans, that made me think that there's, again, the first part of your talk, there's human females that take estrogen replacement therapy. There's human females that take tamoxifen, which to a first approximation is an anti-estrogen. There are women who get overectomized for different reasons. Is it possible to do some kind of retrospective analysis on their bone marrow to see if their protection decreases so that they normalize and look more like males?
Right, yeah. So that's a very interesting point. In terms of CH and some of those therapies, now, I think most of the CH studies has focused on cytotoxic chemotherapies, like such as cisplatin or doxorubicin, and those clearly show association with CH, but things like SERM, or selective estrogen receptor modulators, I don't think there's much has been done in terms of clonal hematopoiesis. we do see some, well, we on a totally different project, we've done some drug screening of modulators of clonal hematopoiesis, and we are finding some surprising drugs that we never thought might affect clonal hematopoiesis, and at least one of them is related to estrogen. So there might be some unknown modulators of clonal hematopoiesis that are not necessarily cytotoxic, and so we're interested in those.
Does anybody else have other questions for Dr. Nakada? I do. That was a fantastic talk. I love this work. And I was just wondering, your strategy so far has been to test the ability of this erythroid biased HSC to respond under conditions of, like, erythroid depletion or platelet depletion. Is that correct?
Yeah.
I was wondering how this interacts with inflammation, and then I guess anemia of inflammation, in the context where you're studying anemia, and then also if inflammation is also a trigger for this part, because inflammation triggers supposedly triggers myeloid biased HSCs, and this being some type of myeloid or erythromyeloid biased HSC, if it also triggers activation of the stem cell, even if it's in the spleen, and how this interacts if you've tested inflammation of anemia.
Yeah, so we haven't studied a particular model other than PHC and bleeding, but we do find that some of the inflammatory cytokines, especially IL-6, is increased after PHC treatment. And so it's really not the PHC is definitely not a pure way to just stress the erythroid lineage. It is causing inflammation on top of that, oxidizing heme and destroying erythrocytes. And so, yeah, there is always a component of inflammation in that PHC model that we cannot really exclude.
Okay, thank you. All right.
I have a quick question, actually.
Please.
So regarding a BRDU experiment, do you have any estimation of how many cell division HSC undergoes in, say, a year?
Yes, so we haven't studied that, but I know Sean Morrison had a paper. Mark Keel is the first author, and sorry for him that I don't remember the numbers, but they estimated the division per year, if I remember correctly. The only thing that I can give you is about 7% of hematopoietic stem cells will incorporate BRDU per day. But I don't have the number of how many division they do over a year.
Okay. Do they undergo faster cell division in a spleen under a PHD treatment, or as compared to the bone marrow?
So after PHD treatment, BRD incorporation is significantly increased in spleen HSCs, but not in bone marrow HSCs.
So does that translate into the more cell divisions, faster cell division?
Right. Yeah. So yeah, we believe so. Yeah.
Okay.
They're dividing faster in the spleen.
All right. If we have no more questions, we'll thank Daisuke for this great talk. Thank you for joining us.
Thank you for the invitation. It's a pleasure.
Yeah, thank you so much. That was wonderful.
Thank you.
Great talk. Thank you so much.
Bye.
Thanks.
Take care, everybody.
Bye.
Bye.